We are a not-for-profit, independent land care organisation with an interest in sustainable agriculture and the broader environment and river health in the Lower Blackwood catchment. You can find out more about us and the work we do on our website, lowerblackwood.com.au. Before we kick off, I'd like to acknowledge the traditional owners of the land on which we all live and work. I acknowledge the Wadandi and Bibbulmun people of the Bibbulmun Nation and pay our respects to elders past, present and future. My apologies to those of you who have heard all of this before, but for first timers, just a few words about how the webinar will run today. You may have noticed by now that you can hear us but not speak yourselves. That's deliberate so we can keep things moving along at a good pace. You can definitely still ask questions though, so please don't be shy. Just type your question at any time into the chat box and I'll make sure it gets asked at the end of the presentation. Also, if you do run into any technical difficulties, please use the status button or type in your chat box to let me know, and I'll see if I can fix it from this end. Don't worry though if you do have to leave early or drop out, as I am recording this, and I'll be emailing out to you either later this week or early next week recording. So to the reason you have all tuned in today, I am tuning in live from New Mexico and USA. I'm delighted to have presenting to us this morning Dr. David Johnson, molecular biologist and senior research scientist at New Mexico State University, and his wife, Hui-Chun Su Johnson. Dr. Johnson has been doing breakthrough work in regards to the efficacy of biologically diverse, fungal-dominated compost for carbon sequestration and improved soil health and crop yields. And Hui-Chun Su Johnson, the woman behind the man, has been an instrumental part of David's research and outreach. Today, they are going to discuss with us the simple and low-cost make on-the-farm tool and method they've developed, the Johnson Su Compost bioreactor system, that can assist land managers to bring their soils back to life. So a very, very warm welcome to you both from us all here. And I will hand over to you to start your presentation.
Thank you very much.
Well, thank you, Kate, for the introduction. Very much appreciate it. And hopefully what I relay today will help some of the farmers and ranchers in that area. I'm here to talk about a biologically enhanced agricultural management approach for regenerating the diversity of life in soils. So the BEAM approach views soils from a systems perspective. It's not just one thing. The compost is one part of it, but what we're trying to pursue is a system, realizing that nature builds everything in systems. And it includes all practices that enhance or improve populations of beneficial microbes in the soil microbiome. So BEAM practices include no or reduced chemical inputs or disturbance from tillage. We'd like to see year-round living roots from full-time ground cover. This is an important component of this to get the energy flow back into this system, as you'll see later in this presentation. You can do this, you can restore the soil microbiology either with the implementation of livestock, with adaptive multi-paddock grazing, or what you would call holistic management there in Australia, or inoculation with beneficial microbes from a Johnson Su Compost bioreactor. So microorganisms have shaped almost every facet of development on this living planet for the last 4.2 billion years. They've accomplished this by doing two things: increasing system biodiversity and improving the efficiency of the capture, storage, and utilization of energy on this planet. You can see the influence of microbes all through the past, where bacteria came in about 4 billion years ago, cyanobacteria, and oxygenic photosynthesis. This is the first step in that accumulation of energy, gathering energy from the sun and storing it and for using it later. Nitrogen fixation, another, microbes originated this. Oxygenic photosynthesis as well. Fungi came about 2.2 billion years ago. ozone layer started to build up, started to see multicellular life. Mitochondria evolved about 1.45 billion years ago. That increased the energy use efficiency 18 times. So all through the past, these organisms have completely changed the dynamics on this planet and made the planet what it is today. So we need to realize that microbes are the foundation of every organism and ecosystem on this planet. You, as a human, the microbiome that you have is comprised of bacteria, fungi, protozoa, and viruses. You have about 10 trillion human cells. You have 90 trillion microbial cells. Excuse me. You have 1 quadrillion resident viruses. So you are outnumbered many times as far as organisms that make up your body. Gene count comparisons. You as a human have about 23,000 human genes. Your gut alone has 22 million genes with the microbial community that's developed there. And your oral microbial genome is about 24 million genes. They do all this for you. They digest your food for you. They generate nutrients you can't get in your diet. They do vitamin synthesis, cell renewal, control your appetites and cravings. They comprise over 80% of your immune system. They turn on and off genes that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. So what happens when these microbial communities are disrupted through either administration of antibiotics, exposure to chemicals, or diet-related issues? We see over the past increases in the usage of fertilizer in these systems. Also, we see an increase in pharmaceuticals and pesticides. Global pesticides have increased 300% since 1970. Along with this, we're starting to see many autoimmune diseases. You can see asthma, type 1 diabetes, multiple sclerosis, and Crohn's disease. Crohn's disease is very debilitating, often lethal, and people can have resections of their intestines to try to cure it. But they're finding that if they bring a fecal matter transplant from a healthy individual, sometimes these diseases can be cured overnight. An example of diet-induced extinction of microbiota is the removal of fiber from the diet of mice. You can see generation after generation, the microbial community diversity decreases all the way down. You say, okay, well, let's just restore fiber back into the diet. Still, it doesn't start to rebuild the diversity that was in that microbial community in the stomach as it was before. And yet when you come in with a fecal matter transfer, it's back immediately. So two things have happened over the last 120 years as a result of our farming management. We significantly altered the soil microbiome with our agricultural management practices. And soil microbial biomass has changed from fungal dominant to bacterial dominant. These two things have created a soil that has lost its biological functionality, decreasing the ability for the soil microbiome to supply necessary elemental nutrients to plants from the soil parent material and our atmosphere. Excuse me. So if microbiota transfers work in the human microbiome, can a simple inoculation of beneficial microbes help restore microbiome structure and function in agricultural soils? Looking at how nature developed these systems, this ladder of succession of coming from a bare soil parent material up to soils that support conifers and old growth forest, you see there's an increasing biomass productivity from 90 grams of dry biomass per square meter in your desert soils up to 2,200 grams of dry biomass per square meter in your old growth forest. You also see changes from bacterial dominant in these low production systems up to fungal dominant. This is where we need to be in this area from about 0.75 to 2.1 for our successional crops, our grasses. But we are currently here. We are bacterial dominant, low carbon, and so our productivity is very limited because of the structure of the microbial community in that soil. We also need to look at the soil food web. Soil food web is very important in these systems. It's the cycling of nutrients. The bacteria have a carbon to nitrogen ratio about 4 to 5 to 1. The organisms that eat them are about 15 to 1. So by the time these organisms get their carbon complement, they have 3 too many nitrogens. They excrete those out into the root atmosphere. That's what the plant picks up. Same for the other trophic levels above that. We also see nitrogen coming from the consumption of living organisms in respiration. See, nitrates are pushed out in the soil as well. So this was nature's system of building a soil in the North America and the Great Plains. The bison were kept in tight herds by the coyotes and the wolves. They were moved along regularly. They only grazed about 40% of the forage that was available. They would deposit their dung, their urine, and saliva. The dung we rolled into balls by the dung beetles, placed underground, basically composted in place or being consumed by the worms in that soil. So this movement of bison across the prairies was basically an inoculation event, and it happened annual every couple of years. So we can do this with cows. We're seeing many benefits of keeping to this grazing pattern of not taking more than 40%, allowing a certain amount of this forage to be trampled down as an energy resource, both for the soil microbes, but also to have the photosynthetic area to recover quickly. If you don't have cows, then we think that we're seeing another way to do this by using a Johnson Su bioreactor. It's a very simple system. As you can see, it's built on top of a pallet. We use landscape cloth and a wire cage to contain it. We put 6 pipe in the middle. These are 4-inch pipe in the middle. You fill the reactor with those pipe in there, and then 24 hours after you've filled it, you can pull those pipe out. So you have airflow up under the pile and up through the center. So it's always aerobic. We also irrigate it about 1.5 minutes a day. It's about a gallon and a half of water a day to keep it 70% moisture. So as I was saying, 70% moisture content. The nice thing about this is no turning. It's aerobic due to those 6 air columns up through the pile. We highly recommend not to let it freeze or dry out. Either one of these will cause it to have to be rebuilt because you're depending on, this is a vermicomposting process. So after the temperature goes below 80 degrees Fahrenheit or 27 degrees C, worms are added. It takes about 1 year to reach maturity, and I'll show you why later. But definitely follow the instructions closely on the first try. No shortcuts, please. And here's a link to how to build it. What we see through the composting process is a change in the community structure from 60 degrees C down to 15 degrees C. You see different phyla of bacteria start out very strong, like the firmicutes here, but they're very small in the population at the end. Others, like the proteobacteria, start out strong, ebb away, and then come back to be a major component. So this is what the diversity looks like in the top 80% of the bacteria in these systems at 4 weeks. You have about 23 species. In the top 80% at 22 weeks, you have 57 species. And this is when most people say their compost is ready. But you can see, if you allow it to mature a little longer, you'll see 99 species. So you're increasing diversity the longer you let this compost mature. This is a slide that shows 2 compost samples made 10 years apart with different materials and yet with the same Johnson Su bioreactor system. You can see striking similarities between the composition of that community. We're also seeing now, when we do metagenomic analyses on these composts, the ability to track the maturity of these systems. And we're working on an inexpensive system to try to be able to test these organisms. But you can see there's 3 composts made in Australia compared to 2 of mine here, and they're almost identical when you look at this principal component analysis on the left. And here you can see that as this system matures, proteobacteria become a major component in this system. So hopefully we'll have an inexpensive system to be able to see how mature these composts are pretty soon. We started out with greenhouse trials with green chili. We wanted to assess the influence of fungal to bacterial ratio on carbon and nitrogen partitioning. In the following chart, you'll see that the root, shoot, and fruit carbon are represented by the brown, green, and red wedges of the pie. New soil carbon is gray, respiration carbon is blue, and all new nitrogen is expressed as the amount of carbon it took to make that nitrogen in these slides. What you see as you go from a bacterial dominant system, 0.04, very bacterial dominant, up to a fungal dominant system at 3.68. We see changes in the productivity of this system. In a bacterial dominant low carbon system, 3% of the energy that plant captured goes into the plant. The rest of it went into the soil, into respiration, into nitrogen fixation. So it must be extremely important for this plant to be able to divert 97% of its energy into the soil to improve the soil. As you move up, fungal to bacterial ratio of 0.84, you see 17%. Fungal to bacterial ratio of 1.6, 21%. And as you go up, 56% of the energy that plant captured went to the plant in a fungal bacterial ratio of 3.68. Currently, in our agricultural system, we're right about here, 11% efficient. So we should be able to increase the productivity of our systems about 5 times. We started out with small plot trials. And as you can see behind, this is where we live in the desert. This is a sandy soil. This experiment was done on a plot of land that has never seen agriculture, so there's no legacy issues to deal with. We were planting cover crops and growing the cover crops, cutting them and hauling them off. So we're just to see what the potential that microbes how they can influence plant growth. We did a one-time application of the compost, as you can see. This is about a 30 centimeter square area. It's just a dusting, but it's about 504 kilograms per hectare. This is our second winter cover crop. Our first one did about 800 grams of dry biomass per square meter. The second cover crop did 1,608. So that's about 540 kilograms of nitrogen per hectare that has come from somewhere because it was not in this desert soil to start with. This is our third year. No fertilizers, again, just biology, and the application of biology was done once. This is a third winter cover crop. We were doing 2,190 grams of dry biomass per square meter. Again, remember, this is a desert sandy soil trial with all the plant biomass cut and taken off after each growth period. This is what it looked like in the first 4 seasons. Started out productivity. The yellow here is our control with no compost. The green is the one with compost. You see almost a doubling of the productivity. But by the 4th season, we were seeing about 2,244 grams of dry biomass per square meter. Now, our winter cover crops, they're planted in fall, late fall. They don't really grow much till about mid-February here. And in 10 weeks, this represents 10 weeks growth. Remember, the most productive ecosystem on the planet is about 2,200 grams. So this winter cover crop produced enough biomass to equal the most productive ecosystem on the planet in 10 weeks. We see a 4.5 times increase in photosynthetic capacity in the treated soil compared to untreated. Now, without the compost, yes, you can improve, but it seems like there's a limit. When you look here on years 1, 2, and 3, without the compost, it never really gets above 2,200 grams of dry biomass for a full year's productivity. And you're up to 3,186 when you have the compost treatment. That's about 872 kilograms of nitrogen per hectare that these organisms fixed. Also, we looked at the metagenome in this to see how the desert profile changed. Our desert soil, you can see not many phyla in this are represented, whereas the control of just growing the cover crops compared to the compost treated, there's 22 phyla out of 39 here that exceed all the others. So you can see the application of the compost does seem to bring back the soil biology. This is what the soil looked like starting on the left here, no A profile. Here is our control. We had about a 3-inch or 7.6 centimeter A horizon. In the compost treated, we had a 17.78 centimeter A horizon. So significantly more penetration of the soil and buildup. We also did some small plots side by side, looking at an extract injected into the furrow at planting compared to an extract applied on top of the soil. This is an image of the extract. It's about a 50-to-1 dilution. In this, we see that when we apply this compost at about 2 kilograms per hectare, we're putting about 900 million bacteria per square meter and about 18 to 54 million fungal spores per square meter. You can see there's a definite difference in the growth. When we look at the difference, you can see the topical application only yielded about 67.83 grams of dry biomass per square meter, where the one with the injection into the furrow yield about 134, about double the productivity. We followed up with the fall in the same plots, doing the same thing, either topical or in furrow. You can see the in furrow, again, over double the productivity. And you can see in the in furrow, you can see it reaches reproductive maturity, where with the topical, it didn't. This is a farmer in Belgium that did a seed coating. And what he observed was double the productivity as well. So this does seem to have a positive influence on your cover crops, starting to rebuild that soil, starting to rebuild that biology. So what do these microbes do for your soil and your crops? Well, we see about 23 nitrogen fixtures, both symbiotic and free living. We see phosphorus solubilizers. Now, if you've applied phosphorus in the past, you'll see that the phosphorus gets locked up due to the physical chemistry of the soil. But these organisms can free that up and make it available to the plant. You'll see organisms that oxidize the elemental nutrients of the soil parent material. You see organisms that put out phytohormone or plant hormones to increase growth. You also see carbon and nitrogen cycling. But what was most interesting in our analysis, we saw quorum sensing. The quorum sensing is when a community of microbes work together and they're able to express genes as a community that they can't as individual organisms. So it's bringing the numbers of those microbes back into the soil so they can work collectively or communally. We did analysis on the metatranscriptome, looking at the messenger RNA, what is being transcribed. There was about over 4,000 messenger RNA in this, and average hits for each messenger RNA was about 62. You see, these here are housekeeping genes. There was about from 1,500 to 2,000 hits, or number of messenger RNA sensed in these systems, but there were two that were all dealing with quorum sensing, and they were from 6,000 to 10,000 hits. So we see a complete change in the dynamics of how these systems work as you bring the biology back. These organisms work as a community to do things that ordinarily don't happen. So as we bring this back into our soils, either through with application of the compost or the proper grazing, this is what we're looking for. We did some small field trials to follow up on that. We did application of the compost on the right here. And after 1 year beam application, we compared it to our control on the left. You can see there's only 50 grams of dry biomass per square meter in the control produced compared to 250. So that's a 5 times increase. And again, no nitrogen or fertilizers were added in these systems. This is year 7. We saw 1,141 grams of dry biomass per square meter. So another 4.5 times increase in the photosynthetic capacity over the 7-year trial. These are some of the summer cover crops we grew in between our winter cover crops. On the left, you'll see black oilseed sunflowers. Normally, they're only about waist high or waist or chest high, but we saw them grow up to 2.2 meters high in these soils that had been rejuvenated or regenerated with soil biology. So we don't know the potential growth that these soils can offer these plants. On the right is Sesbania. The first season we grew it was 1.2 meters. The second season was 1.8 meters. And season 3 was 3.6 meters tall. So each year we can see a change in the growth capability or the photosynthetic capacity of these systems. We threw in cotton crop in 2017 and a chili crop in 2018. When you look at the productivity of these systems compared to the average in our area, we doubled the productivity on both the cotton and chili production. All of this with just biology, no fertilizers. We also saw soil carbon increases over the 4.5 years that we did this. Saw about 10.7 metric tons of carbon per hectare per year put into the top 30 centimeters of the soil. With that increase in carbon, we also saw an increase in the storage and availability of water in these systems. That first 1% increase quintuples the amount of water storage, going from about 90,000 liters per hectare to 450,000 liters per hectare. We also observed a change in the micronutrient availability and macronutrient. You can see manganese and iron here both increased over 1,100%. Keep in mind that our soils are very basic, and these two elements, as the soil gets very basic, are locked up due to soil physical chemistry. And yet the biology has made them available to the plant. Same with magnesium, calcium, zinc, and copper. All of these just by inoculating the soil and growing these cover crops. We also saw increases in macronutrients in both nitrogen, phosphorus, and potassium. Now, these are some of the wilder observations that we've seen, but Paul Transfield in Victoria, Australia, sprayed this on his pasture on one side of this tree. And when this tree leafed out, this area on the left leafed out a full month than the other side of the tree. He was only able to spray on one side because the branch on this side was too low. So it looks like a tree without a trunk. The chestnuts on the left-hand side were twice the size of the ones on the right-hand side. This is Alicia Taft, Rockfront Ranch in California. The difference in the quince that she was able to grow with and without the Johnson Su compost. And Rick came in his vineyard, Johnson Su compost on the left and the organic compost on the right. We're currently on a project with Stella McCartney in Turkey with the company Soctus. They asked us to help them on their cotton production. They wanted to get away from using so many fertilizer and herbicides. We had them come in with the biology, grow cover crops, roll them down, plant the cotton straight into them. You can see the nodules here, the nitrogen fixing nodules on this legume plant in the cover crop. And their productivity was extremely good. They had previously put about 24 trips across this field, either applying fertilizers, herbicides, or pesticides. And yet, bringing in this system, this biologically enhanced agricultural management, they were able to use 85% less fertilizer with no yield impact. In fact, the yields in the beam approach were better than the 100% fertilizer application. So they had huge cost savings on this. The yields were about 4.2 metric tons of cotton per hectare. The crops did well despite government restricted irrigation and low rainfall. They only had 2 irrigation events on this. No herbicides were used due to good weed suppression from the cover crop. They had 100% displacement. They observed biodiversity in their soils come back that they had not seen since their childhood. They've now taken this method to Romania for grain and oilseed crop production. We also do field research on corn. Corn production has the highest land area devoted to crop production in the U.S. Uses about 43% of the total nitrogen applied in the U.S., using 3 times more synthetic nitrogen than any other crop by mass. And what we see when we come in with cover crops, roll the cover crops down and plant the corn, we see significant reduction in the amount of fertilizers, 85% reduction. We see soil surface protection and lower soil temperatures, reduced evaporation, and more efficient use of water. Soil carbon increases as forage for soil microbes and beneficial insect communities, increased cycling and availability of nutrients for commodity crops, higher profits and fewer inputs, and a major shift in these soils from bacterial dominant to fungal dominant. We also recently completed a project in the southeastern US looking at adaptive multi-paddock grazing, which, as I was saying, looks like it's holistic grazing in your area versus continuous grazing. We compared 5 across the fence paired farms on this to see what the influence of AMP grazing was. On the AMP farms, they were able to carry 2.16 times higher carrying capacity than the continuously grazed. Forage biomass production in AMP systems was 46% higher. Soil CO2 respiration was 18% lower in the AMP systems, and soil organic carbon increases were 21% higher. When you look at the effect of these, look like what, small amounts, you see about 9.82 billion tons of CO2 per year pulled out of the atmosphere for that forage biomass productivity. You see about 6.6 billion tons of CO2 per year pulled out of the atmosphere for that reduced respiration, or not put back into the atmosphere due to the soil respiration. Now, the soil carbon didn't add a whole lot to this, but it was about 2.34 billion tons of CO2 a year. If this was applied on 1.25 billion hectares of savannas and grasslands, this would be about 18 billion tons of CO2 per year. That's about half the emissions from fossil fuel combustion. So looking at the soil food web analysis on these ranches, you'll see on the chart here, the green dots are the AMP systems, and the orange triangles are the continuously grazed systems. You see the productivity here of the standing crop biomass in these systems as well. You see the highest producing AMP system had the lowest number of bacteria in that system. You see significant correlation in these farms, as the productivity was very closely related to the higher the productivity, the lower the bacterial count. Although you see the continuously grazed systems are very chaotic. They're dysbiotic, to say there's no correlation at all. We also saw increasing fungal biomass highly correlated to the productivity of these systems. So naturally, the fungal to bacterial ratio, as it is increasing in these systems, is very highly correlated to the productivity. We also looked at a soil food web proxy, looking at all the components, the whole soil food web. And as you can see, as you're increasing the dynamics of the community population of the soil food web, you're seeing higher productivity in these systems. And yet, continuously grazed, again, chaotic, no correlation at all. We also looked at predator-prey relationships. In the AMP systems here on the left, you can see as the bacterial community decreased, you see the protozoa community, which is consuming the bacteria, increase. Very good correlations. You have a very good cycling process going on. You're bringing back all the community members of the soil food web. And yet in continuous grazed systems on the right, no such correlation exists. So Ian and Diane Haggerty, I believe they're close to your area, they do about 20,000 hectares of land, and they're growing barley in their system, but they also graze sheep in between. So they're mixing grazing and row crops. They produce about 3 tons of barley in a region that gets about 1.5 to 1.6 tons. When you look at the influence of the they do an extract, very similar to what I'm recommending, about 2 kilograms per hectare. They do seed coating with it. They do an injection into the soil at planting. They also come back with a foliar application. You can see this seed is just barely sprouted, and yet it is completely developed a soil microbiome around its root structure. What was most astounding to me for them was they had already harvested the barley. In this picture on the left, you can see the stubble from the barley. And they got a late fall rain, and you can see in nice, neat rows exactly where they do the injection was a C4 grass that had not been seen in 4 decades. So this seems to reawaken that soil seed bank as you start to rebuild this soil. Russell Hedrick in the US here in Hickory, North Carolina, he does corn production, dry land. He does full-time cover or commodity cropping, introduced cows, sheep, and pigs into his system. He's been able to reduce his fertilizer applications 80%, and he hadn't used fungicides or pesticides in the last 6 years. His 6-year average for corn production is about 185 bushels per acre. The county average is about 110. He's changed his soil so much that they had an exceptional drought in 2015 with about 4 inches of rain. The county average was 9 bushels per acre when he was pushing out 128 bushels per acre. So he's completely changed the dynamics of what he can grow and how he can grow it in that system, even with little to no rain. Alejandro Carrillo in the Chihuahuan Desert, south of us, south of New Mexico. When he inherited this farm from his dad, it was conventionally managed, and it took about 100 to 120 hectares to support 1 cow. And he's completely changed that with the AMP grazing system. He hasn't seeded anything. He hasn't brought in any fertilizers. He's only done this with cows. You can see he's gone from 110 hectares to 10 hectares to support 1 cow. He's completely changed this, what used to be a desert, into a grassland. Now, you have to consider previously it was a grassland. Before the Spaniard came, there was grass reported belly high to a horse in this area, and all through New Mexico as well. But you can see from overgrazing and improper grazing, it completely desertified the area, and yet he brought this back in a decade. So it's amazing what you can do with animals to bring that biology back. This is what it looked like before. This is an eroded area that eroded in about 20 years. You can see previously there was a 1.2 meter deep soil carbon profile. This, as I was saying, was a grasslands to start with, desertified by overgrazing, and then he has brought it back. So what we're seeing here is a positive feedback loop. As we rebuild the biology in these soils, they support the plant by giving the nutrients. The plants can then produce more photosynthetic capacity per square meter, so more energy captured, pushed down into the soil to feed the soil microbiome. The soil microbiome then extracts the elemental nutrients from the soil parent material or fixes nitrogen from the atmosphere, feeds it back in to the soil system and for the plant to increase photosynthetic capacity again. It's a positive feedback loop, each cycle rebuilding. So we hear a lot of talk about CO2. It's the second most important molecule on this planet, second to water. Without CO2, we would not have the energy that we have today, the energy that we enjoy. All the coal that we've used, the oil, all of this came from photosynthetic operations with organisms. We need to remember that in this process. We also need to consider that when we start rebuilding these systems, there's a lot of estimates as far as how much carbon we can offset. And most of those estimates are done on broken soils. But as we rebuild these systems to see what their capability is, you can see in the AMP systems alone, 18.2 gigatons of CO2, when we put out about 36.8 worldwide. When you start to add in the beam system on the hectares of farmland, that's another 57 gigatons. So that's 76.4 gigatons of CO2 per year. That's over double what we dump into the atmosphere. So there is hope to fix this. And we can see as we align ourselves with nature, we begin to see the productivity of our systems increase, the profitability as well, and reduction of atmospheric CO2 and better use of the water that we get. Below here is a link to my YouTube channel that goes through many of the how to build a bioreactor, how to build an extractor, how to apply it in the field. So thank you very much. I do appreciate your opportunity to present this research, and I hope it's helpful to you in the future for the benefit to your farms and ranches.
Thank you, David. That was just fantastic for us laymen who are not academics and scientists. It's funny, actually.
Oh, you are. You guys are. Every one of you. I've talked to a lot of farmers
scientists
and ranchers.
Farmers are scientists, actually, and they're mechanics and they're engineers and they're business people. They're everything, aren't they?
It's amazing the observations that we get from the farmers and ranchers. They're very astute, very attentive, and we learn a lot. In fact, it's essential that we get to talk to the farmers and ranchers.
Yeah. It's funny, actually, pre-COVID, perhaps most of us wouldn't have even heard of what mRNA is. But now, of course, we all know. And
this is a good one.
Yeah, that's a good one. Before we get to the questions, and there are a few, it just strikes me, I suppose, a comment more than anything else, is that when you're talking about the biology, it's the partnership that's critically important, isn't it? The partnership between the plants and the biology and the biodiversity of those plants.
Yeah, and nature is self-assembling on this.
Yeah.
She doesn't always take the same path, but you can see once you offer the workers, the community, she will choose from those and select and put them to work, basically.
Yeah. Okay, so we've got a few questions here, and Bill asks, back to the slides where you're talking about your trials, how was the compost applied to the small field trials?
The small field trial, it was spread on the ground. It was at that same rate that we observed, was it 508 kilograms per hectare? We spread it right in front of the flood irrigation. So it would go into the soil immediately. Yeah.
And I mean, that's probably fine to do on a small field trial, but when you're talking scale, how practical is that, I suppose?
I wouldn't recommend it. I think it's adequate when you make the extract, inject it into the furrow at planting.
Yep.
Because 2 kilograms per hectare is a good. It goes a long ways.
Yeah, exactly.
But you might have to apply it every year.
You do have to apply it every time when you plant, and be sure that there is, like what David said before, that there is a seed and extract contact.
Yeah.
At a planting, because that you have all the microbes in the extract, and then with the seed contact that they're ready to go to work together.
And we're not sure at year 5 or year 7 or year 10. Do you need to keep applying? Again, it takes time to do this research, regrettably.
And it all depends on your operation as well, because any kind of agriculture is a certain amount of extraction. Any way you look at it, is how much of extraction that you are doing. And for example, when you're doing ranching and you're grazing, that you're not overtaking the forage in able to allow the plants to recover, right? And so when you're doing the row crop, that how much disturbance, how much of plant material taking away, the roots, how much you're disturbing it, or are you able to do no-till? How often are you able to do the cover crop in between the rotation or in between the plants? So all these are variables that you need to really do your good diligent observation of
Yeah.
what you need. So there's no formula for one size fits all.
You
Yeah.
will notice that nature has what I call a golden ratio of how much you can take. And in grazing, it's 40%. And you'll notice in a corn crop, the corn that you take off that field is 40%. The wheat that you take off that field is 40%. The cotton, chili, almost everything that I've analyzed, 40% is that crop that you take off. And the other 60% is there to feed those microorganisms. That's what it takes to rebuild the system and keep it going.
And that's quite conceptually very different from a conventional view, isn't it? You know,
Oh,
leave
yeah.
60% behind? What?
or nothing.
Yeah. And it's very important that, as far as how to adjust that. And whenever you hear about this healthy soil principles of incorporating the livestock and growing the cover crops and doing the no-tills and things like that, but then the devil is always in the details. So when you incorporate the livestock, don't go there and graze it down to a putting green, because your forage is not going to recover. And when you do the no-till, that it all depends on how you terminate. So there are a lot of little details that as a practitioner, farmers on the ground, that you need to take a look at your practice and what will be the most beneficial.
I forgot to mention one thing on the AMP grazers in that study. The longer they have been practicing AMP, or the holistic management, as you said, the more productivity their systems had. It was a linear equation that the longer they had practiced, the more productivity they had.
Yeah, amazing. And sorry, audience, we will get back to your questions in a minute. But while we're on that subject, I just had something that's popped into my mind with the AMP grazing. The trials that you did, were they, how biodiverse were those pastures? Were they, because over here, we have very much an annual low diversity system. In our catchment, there were not originally grasslands, they were forests that were cleared. So we don't have a lot of diversity here. So I'm just wondering that in those trials that you did, was there much biodiversity in the pasture itself to start with, or did that have to be added?
Oh no, this was all just with the cows, just with the grazing.
The forage.
Are you talking the grazing, or are you?
I'm talking the forage. I'm talking the forage itself.
Because, the forage production?
Because we talked about, to have the biodiversity underneath the ground, you also need the biodiversity above the ground, don't you? So in monoculture systems, that makes it very difficult. So you have to introduce more diversity. So
yeah, you've got to have the diversity of the plants, diversity on every scale. Diversity.
So
of the plant.
those grasslands the trials that you did the AMP on, how diverse were those pastures? Were they already quite diverse?
Many of them had been farmed cotton or grains before. Now, they had to be practicing whatever practice they had for a decade. So a lot of those systems had quite a bit of diversity.
Yeah, okay.
But it builds over time. It's not instantaneous. And we're not sure if adding the compost, looking at Paul's results, we were pretty amazed. And he also made comments in there about so wherever he had turned around when he was spraying, the grass was more productive and the cows would go and graze there first. Cows are pretty smart. They know where the good grass is.
Yeah, that's right.
So,
all right, let me get back to the questions then. So Leader asks, what's the best extractor design for large-scale cropping systems?
I've got one online. It's in that link at the very end.
Okay. So
they have a lot of extractors, but it's not something that you want to brew. It's something that made in just a couple of minutes and then is put into a tank, diluted, and injected.
Yeah, and it's exactly like what we said. It's extracting. So basically, you're trying to knock off all those microbes on the organic material and allow them to suspend in the liquid. And you don't want to take too much time and let them sit and brew, because you will bias the diversity and in the species and also the numbers as well.
If you put any nutrients in there, the organisms that can assimilate those nutrients are going to increase in population, where others are going to fade away.
Yeah.
So you lose some of that dynamic range and the diversity when you do that. So just as I say, I have a poor boy extractor online. It's made out of a 55-gallon drum and a agricultural pump. It's very simple.
Perfect.
And you can. It doesn't take very long to make. You know, I can do what, 30 hectares in about an hour or less.
Great. And you don't have to add anything. Just
water.
Just water and compost.
It's just to extract the microbes from the compost.
You just knock that microbes off the compost. You run it through a filter. It goes through the whole process.
I mean, for those who have more resources, there are commercial extractors out there from some companies, and I think some people did purchase those for larger operation. But in our case, it seems to be just fine.
Yeah. And someone's asked about the pH level of the inoculant.
Well, one person answered that already. It depends on the water.
Okay. Yep. And so do you have to worry about the pH though? I mean,
you know, if the compost is made right and if your water's not too bad, no.
Yeah. Okay. So you don't have to sort of watch.
We have high pH water here, and
we're the opposite.
Oh, really? Yours is acidic?
So yeah. So will a bioreactor use wood chips only? Are using only wood chips provide enough food for worms?
If you wait a long enough time, yeah.
Yeah. David did one batch. It was all wood chips, and there were 3/8 minus, which is little tiny wood chips.
That's about a centimeter small.
Yeah, diameter. And it was beautiful, but it took 4 years.
4 years.
Oh,
It's beautiful compost. It's beautiful compost. Dark black.
Yeah.
And it supports worms. And we've seen people pile the wood chips up and it then break down and support worms. But if you want to get this done in a year and have a good compost at the end, you need to use more easily degraded materials like moldy hay, leaves, stall clean out, shred all this up. You want as much surface area as you can get so it breaks down fast. But it is a vermicomposting process.
Yeah.
And actually, when we had our podcast recently, we were talking about the materials, and you were saying how pretty much you start with dried materials, no slimy greens.
Yeah. That if you put the slimy greens, as we call them, in there, they'll go anaerobic on you.
Yeah, and this process is a 100% aerobic process. So if they turn slimy green, well,
you're not doing something right.
No. And one thing I do want to point out is that throughout the process, if you see mushrooms of different color and shapes come and go at different times, it's fine. We have different people report that happen, and it's just natural.
Yeah.
But if you have plants growing out of your pile, something has gone wrong. You shouldn't have anything sprout out of this compost.
Okay. It hasn't gone through that heat phase, obviously, has it?
Yeah, or it has dried out or been frozen, and the moisture content had gone too low, and something had gone wrong if you have a plant growing out of it, or more than one plant.
Yeah, let me say, it doesn't have to reach the high temperatures. It's such an aggressive pile that the microorganisms in there and the microarthropods, they'll devour the weed seeds. When it's done, it's like a paste.
Okay.
When you squeeze it between your fingers, it's just like clay.
Yep.
Yeah,
Yeah.
when it sits there, it looks like a decadent, moist, rich chocolate cake texture. And then when you squeeze it in your hand, it turns into like almost like a Play-Doh. You know, you can squeeze it down and like clay and ooze out between your fingers.
Good enough to eat, you say?
Yeah.
Perhaps not.
After it's transited through a plant, yeah.
Yeah. I think we might have answered Chris's question about how important is the raw feedstock. I mean, you pretty much said you can use anything, but if you use too much wood, it takes a long time, right?
Yeah.
And
Yeah.
Yeah. We only used wood chips when we did some dairy manure.
Yeah.
The dairy manure was very dense, and we put some in just to reduce the density so we could get the airflow in the system.
Yeah. What would be, Bill asks, what would be the best way to apply the extract to perennial pasture?
Well, I guess the way Paul Transfield, he sprayed it. He made an extract out of it and sprayed it on the field.
Yeah.
Or if you're going to do any planting, have any cover crops you try to add some diversity into those fields, then you can inject it into the furrow as you plant.
Yeah, but most definitely should time it with a rain event.
If you can.
If you don't have irrigation, because the microbes definitely don't do well just desiccating out there.
You know, and that's still an area that we haven't had the time. We've been mostly dealing with row crops, and we need to do more research in that area to see, you know, what is the impact of it? Is there an impact on it? Looking at Paul's, it just kind of blew us away to see stuff like that. But again, there still needs to be more research done on that.
But we do appreciate people sharing their observation and their results, because, you know, from what Paul observed, it definitely it supports the theory of that if you have living roots, then it is okay to go out there and spray it topically and living plants. But if you are just planting seeds, you have to inject it in the furrow with a seed.
Either that or coat the seed.
Yeah, or coat the seed.
If you don't have an injection system on your planter.
Well, it's a bit like you were saying, like a fecal implant or for people.
Yeah.
I suppose you're doing the same in the soil, aren't you? When there's nothing going on there.
Yeah.
It seems like that's the way it works. You know, it's so effective for people that with some of these diseases. It's amazing just replacing that biology.
Yeah.
Well, how much difference it can make in their lives.
A few people have said and observed, I guess, that if you've already got good levels of biology in your soil, then adding more is not necessarily you're not going to see much of an impact. But what would you say to that? What would your comments be?
It's possible.
And as long as you're still following, you know, the principles of always trying to have a crop growing on that soil, that transfer of energy, it's all about energy. As we can see now in our societies, it is all about energy.
Yeah.
And it makes an economy or it breaks an economy. It's the same thing in the soil.
And I guess it's as we were talking before, as farmers need to be their own scientists. So you need to go and have a trial area and see if it makes a difference.
Yeah.
Yeah.
Use a small area first. You know, kind of start learning, observing to see what kind of effect you get. But that's the way to do this. You know, you don't want to do the whole farm and change it in one year.
Well, and the beauty of this is that it's so low cost. It's not going to. The only thing it's going to cost you is a bit of time, really, to make it.
Yeah.
And you probably have the waste products sitting around.
Yeah. And why not use all that waste from your farm and turn it into value-added product?
Absolutely.
And that's just totally closing the loop of the circle there.
Catherine says, I have a beautiful 15-month-old IBC reactor made from almost pure eucalyptus leaf litter. It's only just starting to collapse down and is still very active under my microscope. How do you estimate the amount of material that will be produced by a single reactor depending on the biomass used?
Okay, the first thing I would say is 15 months and it's just now starting to collapse. Normally we see that reduction of, about 4/5 of that, or no, 3/5 of that reactor will settle down into the 2/5 volume in a year. And it happens relatively quickly after the 6-month period. So I would suspect that, maybe she doesn't have it wet enough. It's not biodegrading fast enough, or the materials she has in there are not biodegrading. The second part of the question was what now?
How do you estimate the amount of material that will be produced by a single reactor, depending on the biomass used?
I like to look at it through the microscope and see what the population is. I usually do a 50-to-1 dilution, and I use a hemocytometer. It's a little gridded microscope slide that the smallest squares on there are about 0.25 nanoliters. So you can do counts of bacteria and counts of fungal spores. And if she would go by that one slide, I think it was 15 to 64 million fungal spores per square meter to 90 million bacteria. That would be something to shoot for.
But as far as the volume goes, it's probably only about 2/5 of the volume at the end, because typically we started out with a wet material, built about 2,000 pounds.
Well, we'll have to do it in pounds. I didn't do the conversion on that.
Sorry about that. 2,000 pounds. And at the end, we ended up with about 700 pounds.
Yeah. In that 5 foot tall by 4 foot diameter bioreactor.
So you ended up with about 2/5 or so, somewhere around there. It depends on how dense or how loosely that you fill it. If you fill it too dense, you might end up with anaerobic condition. But then if you put it too loose, it's got too much loft to it, then you might not have too much left at the end, because it will be all digested.
And I remember, eucalyptus leaves could be a bit like wood chips. Perhaps they just take a lot longer because they've got that coating on the top of them.
One of the composts in that analysis, the metagenome analysis, is from Australia. Oh. It's from Terry McCosker, and he used eucalyptus leaves, and his was the best compost of all of them.
Really?
Compared to ours.
And there's actually 2 commercial products.
Yeah.
Compared
and
in there.
So you do have one, at least that we know of, a very successful Johnson Su Compost bioreactor builder there, Dr. Terry McCosker.
Well, it's very encouraging to think, because we've certainly got no shortage of eucalyptus leaves. If we can use those, that is fantastic. Yeah. But I also remember in your podcast saying to me that your bioreactor design is circular because of the distance for the air to be able to get into the compost, and the IBC reactor is a square vessel. Would that have an impact?
I think what you need is IBC tote. They're square, and what I would really like to see is that they actually use the woven landscaping cloth to line the outside, because this is aerobic composting process, and that woven landscaping cloth is not just holding the material in, it also allows the permeability of the air to infiltrate through the pile to allow it to stay aerobic, but also the density of the material allows the pile to stay moist all the way to the edge. So that's why we try to caution people not to substitute the building material. Shape-wise, if they would put the those air pipes in certain distance, so that way the air can infiltrate through, any beginning edge, no more than 1 foot to the other one, where the air can start to infiltrate through.
That's about where we noticed in the windrow composting that air only penetrates about 1 foot into that system, and then it goes anaerobic. And that's why I have to turn it so often. Otherwise, it's a lot of odor.
Mm-hmm.
And then that's how we design the bioreactors, never be more than 1 foot away from ambient air. And
Okay.
can you see this slide, or can they see this slide or not?
Yeah, they can.
Okay.
Yep.
This is at a 50-to-1 dilution, what the compost should look like. You can see these are spores here, 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 and here. And then the bacteria are these
Little tiny
the
ones,
little white
dots.
So, and that's kind of what you're looking for.
Yeah.
In there, about 50 bacteria. This one actually has more than that. I think it's about 100. When you zoom up, change the focus area, you can see there's a whole nother layer of the same thing. So there's a significant number of bacteria, and this looks very good as far as the fungal spores as well.
So I would say that anybody who are interested in building the compost bioreactor for their farm, and it might be great for them to invest in a nice microscope, and they can start practicing looking at not just the compost, but also their soil. And they can start learning and study about the changes.
Yeah, this one is turning clusters here. And this is all increasing maturity that we're seeing. And hopefully we can develop this into about a $40 or $50 test to be able to tell the maturity of the compost.
And just for our audience, if you're local, we have a microscope you can use at the land care office. Just need to give us a call. You can bring your sample, and happy to let you use it. I suppose we need to get moving on with these questions. There's quite a few. What do you do if you have soil that was tested as totally absent of a mineral, in our case, copper?
Now, my first question was the soil test just soluble copper, or was it a total elemental analysis to see if there's copper that's just not soluble? And that's most likely the case. Very few people will do a total elemental analysis on their soils. I recommend it. So you get an idea of what is there. Now, whether it's soluble or not and available to the plant, that's another question. But the microbes, as you can see, they increase the availability of those nutrients. They can actually extract those elements out of the soil parent material in concert with fungi. The bacteria work on it, the fungi work on it, and then the plant just gives them the energy in order to do that extraction process.
Yeah, a whole ecosystem approach.
It's a beautiful system if we just allowed it to work.
Yeah. And Sarah asks, does the process of composting, I'm guessing, need to be kept cool? We have above 40 degrees summer days, so should we place it in a shed?
No, we see 43 degrees C regularly here in the desert, and as long as you keep it moist, if you keep that 70% moisture content, you're good to go.
And you do need to put a piece of a woven landscaping cloth over the top and secure it.
That'll keep the moisture in.
Yeah. So it keeps the constant moisture in there so it doesn't dry out on the top, but also you can keep the birds and other wildlife from getting in there too, to eat your worms.
Well, the rats who like to get a bit of shade.
Yeah.
Now, I would have noticed the sprayers on this system. This is called a spit sprayer. I'm sure you have them in Australia, but they put out about half a gallon a minute or less, and then I have about 6 of them, I think, inside. So it's spraying a mist on the system. So you just mist, and it settles on the compost.
Yeah. Someone's asked, or David's asked, do you screen the compost to get the worms out before application?
No.
You
can.
They're sacrificed.
They're sacrificed.
Actually, the way that the extractors works, I'm swishing it around with my hand, and they do survive that process. So in the compost that's left over after you've washed the bacteria and the fungal spores off of it, they do survive that. So you can.
Not all of them.
Not all of them.
Yeah. Oh well. And could you make this compost without worms, or is it recommended to use them?
I think it's essential. I think vermicompost there's so many factors. A worm adds to this.
Yeah.
Growth factors on the plant, completely different microbial communities come out the back end of these systems. There's so much we don't know about this, but we do know that worms are essential. And you see this as you bring these soils back. You see it when you're grazing properly. Those soils have worms.
Yeah.
Yeah.
And that's part of the Great Plains. That's why we had soil carbon profiles 6 foot deep or deeper.
Mm-hmm. Yeah.
And we've just been mining those for the last 150 years.
And Catherine asked, can it speed up soil recovery after fire?
I would think so, because you get quite a bit of damage to the that whole soil microbiome because of that heat. And that's part of nature's rebuilding. She'll tear things down and rebuild. So and she has that capability, but it all seems to come back to the soil biology.
You would think so.
I have not done the research to do that, but I would expect, yes, you would see significant.
I'm going to try on. We did a couple of autumn burn piles, and of course, everything's dead underneath now after the burn pile. But I've got some Johnson Su just ready to go, so I'm going to go and try. So, Catherine, I'll let you know.
I love citizen science.
Yeah, we love it. Please do share the information. We can learn something.
And that's another thing. It's really good if the farmers and ranchers can work as a community.
Yeah.
Take the hint that nature has in microbes working as a community to do things that they can't do as a single organism. And as a community of ranchers and farmers, each one can try something different, and everybody can benefit from that research.
Yeah, you're so right. Shorty asks, can you inoculate new bioreactors with compost from an old bioreactor?
I always do. I throw it into the bath water. I'll grab a handful occasionally of the mature compost and throw it in the bath water just to make sure.
Yeah. So that actually is a good segue into Miroslav's question. What is the recommended moisture content of compost below which it must not fall during composting?
70%. This is ideal for both the worms and fungi. That's where they really shine. And that's why you have such a fungal dominant compost coming out of this when you compare it to other compost.
And Catherine, she's in love with her compost. Of all the compost I've looked at, my JS is gorgeous. Lots of sexy naked amoeba and fab diversity. I've taken a lot of care to make sure it's aerobic. I use landscaping compost. So Catherine is the one who's got the eucalyptus, I think. Yes. So she's previously made other compost as well, by the sounds of it. Chris asks, you had a slide that said, if this is your first one, follow the directions, no shortcuts. Can you elaborate on some failure modes?
Oh, how much time do you have?
Maybe just the top 5.
We've seen them use screen that was too open, and the water would just shoot out the outside and the pile would dry out. We've seen them freeze. I think that's the two biggest things, is just drying out and freezing.
And also they people who skip the soaking and draining process, and they ended up with a lot of sand in the pile. And if you think about, you're only using 2 kilo per hectare. So if majority of a weight in that 2 kilo are going to be sand, it's not going to be the quality that you need to inoculate your hectare.
The water bath settles all the sand and rocks out of it and floats the plastic, if there's any, to the top. So you really go in with a lot of just plain organic matter in the system.
And also it does not allow it to compact as well if you have a lot of rock and sand in there.
And it's a good workout.
Yes, it is. These are not easy to put together, but you don't have to turn it.
Yeah,
that's right.
One thing I do want to remind everybody, before you start it, of course, you need to find a spot that you can get water, set up, and hopefully you put a timer on it so you don't have to go out there and water by hand. And make sure that spot is flat. It's level and firm. Because we know somebody that they made sure it was flat, it was level, but they didn't realize there were what was it?
Gopher
holes underneath it. And
we don't have that problem here.
Once it starts to tilt,
yeah,
it doesn't stop.
Because when you start filling it, it's very heavy and it collapses the gopher hole.
Oh,
so make sure that it's level and firm.
and also put plastic underneath the bioreactor, underneath the pallet.
Oh yeah.
Because if a tree's close, the roots will find this pot.
into it. Yeah, actually, and that's well, I think Sandy, they might have answered that question. It doesn't really need to be in a sheltered spot under trees, does it?
No.
I mean,
or rain. Don't worry about rain either.
Yeah.
You can apply as much water as you want to these things, and it'll just drain through slowly. You know, it doesn't waterlog because it's all organic matter, and it has the microorganisms in there, and the worms always making holes, microarthropods making holes for air and water infiltration.
Yeah, and actually my piles, the actual hole, the columns where the columns were collapsed a few weeks ago, but it's right down to it's only about.
Yeah, you're looking good then. They're supposed to collapse. Yeah.
Yeah, as long as it's not premature, but at the point where you are, that is fine because you got.
No, it's about 9 or 10 months old now, I think.
Yeah,
probably.
It just happens naturally. You know, they'll just close in on themselves and turn into that decadent, rich chocolate cake.
Oh, I'm excited about breaking into it. Well, not yet. I'm not. I'll wait just a little bit longer. Sonia says, are any of the microbiometer testing kits any use for getting a starting point for your soil fungi bacteria ratio?
I try to shy away from dissing other people's products, but
we bought one and tried it.
I'll tell you, the screens they have are great in them, and the little mixer is also great for doing the analysis that I do, you know, through the microscope. I really recommend, if you can look at it through a microscope, do that.
Tell them why.
When I did the compost, you know, I did some analysis. I did serial dilutions to see if things stayed the same as you diluted it, but no. The fungal to bacterial ratio should remain constant, and it didn't when I did different dilutions of the compost. So it depends on how much material you use. There's a sweet spot that it works in, but it's too narrow to see any dynamic range in the efficacy of the compost or the condition.
So the short answer is, for us, no, it's not useful.
Well, actually, most sheep farmers, or many sheep farmers, would have a microscope anyway for doing fecal worm egg counts. So
yeah, and if they've got that, a hemocytometer is not that expensive.
Yeah,
and you can learn so much. It's fascinating just to look at the soil under a microscope. It gives you a whole new perspective on how these systems work.
Well, also you've put all that time and energy in. You want to actually see what the outcome is, and know what worked.
That gives you an idea of what rate you need to mix it at. You know, if it's a really good compost, you can get down in that 2 kilograms. If it's not,
you might need to use more.
Yeah.
That's a good point. Anthony asks, if you live in an environment that regularly frosts during the winter, how do we stop it from freezing? You
know, we have frost as well. We've gotten down to, well, you're going to have to do the conversion, Anthony, 17 below in Fahrenheit.
But that was a freakish event, though. That's not a good example.
It will survive a night or two of freezing, but if you're in a climate where it freezes hard,
and constantly,
it never gets above 0, that might be an issue.
You
need to indoors or in a root cellar or in a hoop house. You can weather a certain amount of it in a hoop house.
Be creative about keeping it alive and not freezing.
We've had people in Quebec do it at 45 below C. So
I'm not sure if you're going to check it when it's that cold.
Forget the worms, huh?
Yeah.
But they create an environment that they are able to keep everything from freezing in a greenhouse of some kind of system that they hook up.
Yeah. Shorty asks, are mites normal in a JS bioreactor? I found these mites in my last bioreactor and they are helping to degrade the complex lignified materials. Yeah,
they're part of the biodegradation process as well. You'll see all kinds of things in here. See all different kinds of fungi. They'll put the fruiting bodies out, and we've seen red, we've seen yellow, we've seen pink.
Yeah, we have people email us and send us pictures, and they say, this is very strange looking. And, I got this kind of pink foamy slime or something like that. They were a bit worried, but then it disappeared after a little while. Yeah.
Bill asks, is chopped lucerne hay still high on your recommendations for what to fill the JS reactor with?
Yeah, I've had good luck with it. I do seem to see that if it's hard to keep wet, then it's because of all the stems in that lucerne. You might be able to add some leaves or stall clean out to it, or grind it up a little finer. If you have a bale grinder, run it through a bale grinder or a chipper shredder. It works for me, but I've had a little issue around the edge drying out because the stems don't degrade fast enough, and it'll dry out on the edge.
Yeah, I always find that I will get a little bit of top drying out, just around the top.
Yeah, top is okay, but if you're getting the whole side of the bioreactor, you probably have to redo that material on the next run. I mean, it'll still biodegrade in the center, but you don't get that rich chocolate cake.
Yes, that's what you're after. And Chris says, what is your indication for when to add worms?
You want it to be, let's see, below 27 C.
As soon as you put it together, the temperature usually is starting to rise. And then you kind of need to check on the temperature that it will reach a certain temperature, hours. It varies, depends on the material that we use.
65 degrees C. And it only goes there for about 3 to 4 days.
Typically.
And then it'll start coming down. It'll be through with the thermophilic phase normally in 3 to 4 days.
And then after that, once it comes back down and reach 80 degrees Celsius, not Celsius, Fahrenheit, 80 degree Fahrenheit. And that you can put the worms in safely.
If you can stick your hand in there and it's not too hot to keep it in there, then you're good.
Well, it looks like we're the end of our audience questions, but I have just a couple more I want to ask. Was you talked in your slides about adaptive multipace, using livestock, and in the absence of livestock, this is the JS compost extract is an alternative. What about and instead of or?
Well, yeah, we'd like to know that.
Okay.
That's got our curiosity up as well. But to do anything in agriculture, it takes 3 to 5 years, and with the funding that we get, we're very lucky to have gotten this far in the cropping. So it's in these projects. That the project in the southeast, that's about $400,000.
Wow.
To get all that data and get it processed, and this stuff is not cheap.
The lab analyses are expensive.
But I'd love to hear what you find out. Maybe if we see some funding come through, we can tackle it ourselves.
Yeah, I guess we're obviously doing demonstrations but we're not doing scientific trials. But we're doing projects in our catchment with trying to put more diversity into our pastures, moving away, putting more perennials and obviously more different species in there. And at the same time, so that means seeding activities. So when we're seeding, we can put in, we can seed coat with JS extract, and we can also put in furrow but those pastures will also be grazed. So
Yeah,
I guess we could do some trials where we don't do the grazing, just the biology.
Yeah, you could do some exclusion fencing and see what happens.
Yeah, and see what happens.
You know, when I started on this, it was all towards addressing CO2. And I looked at what areas had the most impact in cropland, since it's a much more controlled environment, was that area. And it just happened that what we stumbled on worked really well in cropland too. I mean, a lot of this is just serendipity.
Yeah. And I suppose the same principles apply. If we're trying to improve the biodiversity in our paddocks, we need to and because we don't have native species going to be coming through, even with grazing. So, you know, we need to add that diversity. So we're going to have to seed.
You might be surprised at what comes back. You know that.
Yeah, there'll be some for sure.
Yes.
There will be some, but if we want to speed things up a bit, we can add some in. Yeah. So if we're going to seed, then we might as well seed with some JS extract while we're doing it.
Yeah, you're there. You might as well. And like I said the other day about plantains being good for worming the cattle, because if you're adding ivermectin to the cattle to keep them dewormed, that just kills the process.
Yeah, you kill the microbes.
If those patties are not disappearing in 24 to 48 hours, you do not have the
beneficial,
the dung beetle populations that you need in order to keep this system cycling. So it's a really good indicator of the healthier system is how old are those patties.
Yeah, you want to get to the place where you're regenerating is that increase that the rate of cycling and efficiency of cycling.
All that plays right back into the photosynthetic capacity of this system, and that's what it's all about. Is the capture of that energy.
Yeah.
Well, actually, and I don't know if you are available for other emails, but Shorty has asked, what's the best email for us to email questions to you? I know you're very busy, but I still have a few more questions.
Yeah, just be patient, but it can be either John Soda, JOHNSODA at NMSU.edu. That's New Mexico State University. Or just David C. Johnson at NMSU.edu.
I use the other one because sometimes that second email get bounced for some reason.
David,
be
patient,
happy, please.
Well, when I email out the recording, are you happy for me to let people know your email address then for that?
Yeah, that's fine. And you know, it was on the bottom right of the slides, but it disappeared in the transfer.
Yeah.
Kind of odd.
Well, I think we might come to the end, and pretty much right on time, just a couple of minutes to spare, and that was amazing, of course. And it seems to me, that such a simple and low cost system, really, which brings about such phenomenal results, is a bit of what we call a no-brainer, really. Why wouldn't you have one on your farm? Why wouldn't you have a go at making one? It's a bit of time and effort, obviously, to build the bioreactor and put the material in, but then you don't have anything to do for a year and wait and get this beautiful thing at the end. So I think it's worth the investment for sure. And for any of our audience that are locally based, we are actually running a hands-on bioreactor workshop this week on Thursday. David and Hui-Chun will obviously not be jetting in for that, but they have offered to run a special Zoom Q&A session for participants sometime after the workshop, a date yet to be decided, to cover any questions that might come up once you've actually had a go of doing it. We still have a few tickets left, so if you're interested, please head to our website to book. But once again, thank you very much to you both for your time. I know you'll be very busy people and very much in hot demand, so we very much appreciate that you were able to come and talk to us today. And we'll let you go now. It's getting a bit late and you can go and have your dinner.
It's our pleasure as well to see everybody interested, and this seems to be a good path forward. It's more profitable. Definitely.
We wish you all success and share the information with us and with everybody else, and so we all can be successful.
And for our audience, that's it for today. But before you sign out, Can I ask you to complete our webinar evaluation form? It'll only take a few minutes. It's super helpful for us in making sure we are providing the content that's important to you. Oh, it's just dropped out again. Sorry about that. So if you wouldn't mind taking the time to complete that, that would be marvellous. And many thanks to you all, and enjoy the rest of your day.